Hello friends, welcome to my channel Naved Biochemistry Classes, and in this video I will talk about mechanism of splicing mediated by Spliceosome. In previous lectures and videos I have already discussed formation of Spliceosomes. Okay, then I also discussed something related with mechanism in previous video. Now let us see, we will start in this lecture the steps involved in formation of a spliceosome, and then how finally the exon is joined and intron is removed and a spliceosome disassembled when intron is removed along with as a complex associated with spliceosome. So what happened for this spliceosome formation? The first step, obviously, when you say transcription, when you say transcription, you are saying that mRNA is being synthesized from DNA. So when this mRNA containing spliceosomal intron is synthesized, you can see here the structure here. Not a structure, rather a graphic representation or just a representation. You can see DNA. Now the mRNA is being synthesized. This is the 5' end, this is the 3' end. The green are exon and the red part is intron, and you have that is a spliceosomal intron. And of course at 5' splice site you have GU dinucleotide, and at 3' splice site you have AG, and you have this adenine branch site that is containing adenine. We have already discussed the composition of spliceosomal intron in our previous lecture. So actually what happens, it is not as such that these are synthesized in a stepwise. So suppose here we will just consider that in the synthesized mRNA precursor, it is synthesized up to adenine branch site. So what will happen once the synthesis start of mRNA? The snRNPs, SNRPs get associated with the according to their specificity towards the nucleotide sequence of intron. So now what is the first step of a spliceosome formation? So you can see that a spliceosome formation begins with, you see here that UN is being bind to 5' splice site, that is dinucleotide G. So spliceosome formation begin when UN SNR bind to 5' splice site of mRNA precursor. You can see here. Now, obviously, for this binding, UN SNR has a sequence near its 5' end. So here also, when we say SNRs, we are talking snRNA associated with protein, and when you say snRNA, you are talking a small nuclear RNA of 50 to 100 nucleotides length. So 5' end of this UN SNRP has complementary sequence as compared to a splice site of at 5' end of the intron of the you say intron, or since we are typing spliceosomal intron, so it is a spliceosomal intron. So what is the next step? Next step is attachment, or you can say binding of U2 SNRP, where it binds at the branch site, that is at adenylate residue. Okay, and you see here as a result of it, the binding of U2 SNRP and the adenine of rather adenylate residue of branch site results into formation of a stable complex. And it has been found that this stable complex contains 40 nucleotides. But here you can see the process is written, and you can see 1 ATP is required. ATP is required for this process. Now base pairing, here the base pairing occurs between U2S nerves and adenylate residue of the branch site. So what will happen? A bulge will be formed. That is, it will be pushed. That adenylate residue will be pushed. You can see a bulge is being created, and this binding also leads to activation of this adenylate residue as a nucleophile. So after this combination, adenylate is ready to act as nucleophile. Now, as the transcription grows, as the mRNA synthesis grows, so what will happen? The 3' spliced site will also be synthesized. That is, it will come out. As soon as 3' spliced site is synthesized, what will happen? U5 will react with, rather it will bind with the 3' spliced site, gets associated, and followed by U4 and U6. Followed by U4, U6, SNRP, SNRs, which bind to form Spliceosome. Okay. Now, so here, once you have to only, it is a bit specific that U5 will attach to 3' spliced site when it is synthesized during transcription, which when it comes out, and it is followed by binding of U4 and U6 with this what is being developed, that is a spliceosome, and all together this binding will form a spliceosome. But this is inactive. So what is being formed by this all combination? An inactive spliceosome. Okay, and you already know that when we talk spliceosome, it is made up of 5 fundamental SNRP, that is U1, U2, U3 is not there, U4, U5 and U6. Well, once inactive spliceosome is formed, it will be converted into active spliceosome by expelling 2 SNRPs, that is U1 and U4. That is of the complex which has been assembled to form the inactive form. In the next step, U1 and U4 will be expelled from the complex. Okay, and ATP will be required for this process. So what will happen again, and in this step also, what happened? U6 SNRP binds both with the 5' spliced site and with the U2 SNRP. Okay, so to make to convert an inactive into active spliceosome, the U1 and U4 SNRP will be expelled, and U6 will be pairing with both U2 and the 5' spliced site, and obviously ATP is required for this process. In the next step, lariat formation takes place. That is the shape of the rope which is used for catching or tying the animals. That shape will be formed. So you can see here the active spliceosome undergo lariat formation. How lariat formation takes place? Actually, what happens? The branch site adenylate OH, as I told you, that due to when U2 gets attached to adenylate, it pushes it, bulge is created, and it also activate the adenine or adenylate residue as a nucleophile. OH of this adenine attacks the guanine nucleotide at 5' exon splice site. That is here 5'. So obviously here it is 5'. This is exon, so you call it 5' splice site or 5' exon splice site. And what happened? Due to this nucleophilic attack, a bond, phosphodiester bond will be formed, but it will be 2', 5' phosphodiester bond. Between whom? Between adenine and guanine nucleotide, a bond will be formed, 2', 5' phosphodiester bond will be formed, and this will lead to cleaving of guanine residue. Guanine residue will be cleaved from mRNA strand. It will be cleaved. Okay? And all this process obviously is involving a trans-esterification reaction, and this is the first trans-esterification reaction, as I told you earlier that during introduction part, that two of the mechanisms that the splicing involves two trans-esterification reaction and Mediated formation. So here is the first trans-esterification reaction. Now further what happens, once guanine is cleaved, you have exon, 5' exon having free 3' OH, and this 3' OH group then attacks 3' splice site of branch radiator structure. Here you can see this is branch radiator structure. This attack is taking place, and this step also involves a trans-esterification reaction. So this is the second trans-esterification reaction, and finally what you can see the two exon, 5' end and 3' exon being joined. The adjoining exon is now covalently bonded, which was the target of splicing by spliceosome, mediated spliceosome, and intron is removed along with the spliceosomal complex containing your U2, U5 and U6 SNRs, and this disassemble. Once intron is removed, the spliceosome disassemble. So the final result of splicing mediated by spliceosome will be having a covalently bonded adjoining exon. That is, you can see this exon and this exon will be covalently bounded, and this excised, that is the which has been cut, you call it excised intron, and obviously this is the spliceosomal complex. You can see here in a Mediated form, it will be released, and this Mediated form spliceosome having intron will be having U2, U5 and U6 SNRs, and it will be released, and as soon as intron has been removed, that is the process is complete, the whole this complex disassemble, that is spliceosome disassemble, and it is liberated. You can see U2, U6, U5 and U6 SNRs will be. So once the intron has been removed, the spliceosome will disassemble, and obviously the component of it will be released in the nucleus, that is your U2, U5 and U6 SNRs. Now it has been found in some rare mRNA. What happens? You don't have GU, you don't have AG at the terminal. You can see 5' site and 3'. So what is there? It is 5' AU. Here you will have AU. In some rare you have, like if you have 5', then you have AU, and obviously branching of adenylate, adenine will be there, branch side, and but here at 3' you will be have with AC. So you have now AU in its place of GU and AC in place of AG in short and rare mRNA, and in that case splicing, here U11 will come and here U12 will come. Okay, that is U12. That is U1 and U2 will be replaced by U11 and U12, and this is the statement, that is some mRNA precursor intron are removed by U11 and U12 snRNP, which replaces U1 and U2 snRNPs. As you know, U1 goes to 5' when it is GU, and U2 goes to adenine branch site in normal mRNA. But here if you have AU, you will be, U1 will be replaced by U11 and U2 will be replaced by U12. Okay, and which mRNA? Yes, specific, you have a nucleotide which determines this. The nucleotide is at 5' you have AU, and at 3' you have AC dinucleotide. So you can see that this is all about splicing mediated by spliceosomes. So thank you very much for watching my channel. Please subscribe.